Dear colleagues, as we unfortunately had to cancel this year's World Congress in Montreal, we thought it would be a good idea to at least share some highlights from the virtual Congress that took place in February. Before the video starts, I would like to announce that the 2022 World Congress in Taipei, Taiwan will take place from June 9th to the 12th. I look forward to seeing you there in person, and now please enjoy the video. Oh, and don't forget to subscribe to our YouTube channel below for free so you can watch more videos in the next weeks. Thank you.
So my talk today, as I mentioned, is abnormal core clock genes and MDD, potential resetting with rapid-acting antidepressants. I'm from the School of Medicine, University of California, Irvine, USA. So this is clearly a global health problem. The World Health Organization ranks depression as #1 of all medical diseases in terms of lifetime disability. Depression is associated with a high incidence of suicide. WHO reports over 800,000 suicides per year worldwide. In the United States, the suicide rate over the last 20 years has increased from 28 to 48,000 per year. Circadian manipulations can induce striking treatment responses in a subgroup of mood disorder patients. These include non-pharmacological, non-invasive treatments, sleep deprivation therapy, bright light therapy, sleep phase advanced therapy. These therapies all alter circadian rhythms. So rapid-acting antidepressant response to sleep deprivation therapy is low-dose ketamine compared to standard antidepressants. On the bottom, you have weeks. On the left side, you have percent of improvement. Standard antidepressants take 5 to actually 10 weeks to produce full response, while low-dose ketamine and sleep deprivation produce a response within 24 hours or more rapid. Methods involved analyzing highest quality brain tissue, all with rapid death, and most important, accurate time of death, high pH and RIN values, and short BMI. Microarray results of 12,000 transcripts were screened for genes having a significant 24-hour cycle. We evaluated the statistical significance of the findings by permutation, randomly reassigning time of death data across subjects 1,000 times. Brain tissue from 34 MDDs were compared with 55 control subjects. And here on the top bars, you can see controls, and the bottom MDDs. You see 6 brain regions along the top and 5 core clock genes along the left side. The light blue indicate significance, and you can see a large number of significant boxes here for the controls, and the opposite for MDDs, which the dark indicate non-significant. The anterior cingulate, ACC, is a key pathophysiological brain region in MDD. 20 out of 26 studies showed that increased ACC activation at baseline predicts antidepressant treatment response. Significant dysregulation of the top 12 circadian clock genes in human brain, MDD, match control, in the anterior cingulate. And you can see the match controls, only 1 was not significant, while in the MDDs, only 1 was significant. Extremely interesting result. ACC has extensive projections to multiple brain areas, thus disruption in clock gene expression in the ACC could account for the profound changes associated with depression, which I've mentioned before, altered hormone secretion, temperature, mood, and sleep rhythm. So these are 5 circadian-related genes, downregulated by clock genes. The blue indicates controls. The red, ketamine, and the green, sleep deprivation. You can see in all of these cases, they were significantly down compared to controls and concurrent. Putting this together, ketamine, sleep deprivation interacts with the ACC, common circadian transcriptional pathways, and rapid antidepressant effects. So in summary, Major depressive disorders, MDD, have abnormal circadian rhythms, sleep, hormonal secretion, temperature, and mood, all of which are controlled by clock genes. Evidence for altered clock genes across 6 brain regions in MDD compared to controls are presented. There is evidence that the anterior cingulate, ACC, is a critical brain region in the pathophysiology of MDD. And of the 6 brain regions, core clock genes are most significantly altered in the ACC. And finally, there is a common genetic signature for ketamine and sleep deprivation, with 5 circadian genes significantly decreased in both in the ACC compared to controls. We show that clock gene machinery is altered in MDD and hypothesize that resetting abnormal clock gene machinery may make a major contribution to the mode of action of the fast-acting antidepressants, ketamine, and sleep deprivation. Thank you.
Hi, my name is Colleen McClung. I'm a professor of psychiatry and clinical and translational science at the University of Pittsburgh School of Medicine. And today I'm going to talk about strengthening of circadian rhythm amplitude, how this stabilizes and improves mood, and mostly I'm going to talk about evidence from animal models. And we know that circadian rhythms are associated with psychiatric disorders for a number of different reasons. First, we know that shifted and blunted rhythms, as well as a late chronotype, so being a night owl versus a morning person, are associated with psychiatric disorders, including bipolar disorder, depression, schizophrenia. We also know that environmental changes to circadian rhythms, like shift work, jet lag, seasonal changes, can trigger or exacerbate mood episodes, particularly people who have bipolar disorder. We also know from a number of human genetic studies that polymorphisms in circadian genes, these are genes that make up the core molecular clock, are associated with a variety of psychiatric disorders. And as well, there have also been some genome-wide association studies which have linked polymorphisms in circadian genes with particular psychiatric disorders, in particular bipolar disorder. And we know that targeting circadian rhythms can be therapeutic for certain individuals with mood disorders, and these include things like bright light therapy, interpersonal and social rhythm therapy, for example, things that will amplify and stabilize or shift the clock in a particular way can be therapeutic. So circadian rhythms are controlled by a master pacemaker, which is located at the base of the hypothalamus. This is called the suprachiasmatic nucleus, or the SCN. The SCN is directly responsive to light input that comes in through the retina. And then that light input will synchronize cells in the SCN, which works similar to the conductor of an orchestra, where it controls the timing of the rhythms that go on throughout the brain, throughout the body. So while there's a clock in nearly every single cell in the body, it's really the SCN that is kind of that conductor of the orchestra that keeps it all in sync, keeps it all rhythmic in time. So we've been studying this phenomena in animal models. And one of the animal models we use is the unpredictable chronic mild stress model. And this is a model where you put mice through very sort of minor stressors, but the trick is that they're unpredictable, they're random, and they're chronic. So things like forced bath, wet bedding, aversive smell, restraint stress, things like this. And after several weeks of doing this, the animals develop what we call a Ucms syndrome, which is very similar to depression, at least as similar as you can get in a mouse. So they have increased anxiety-like behavior, increased sort of depression-like behavior or behavioral despair, anhedonia, so decreased pleasure in things like food. And we use this as a model to be able to look at the effects of chronic stress on measures of circadian rhythms. So when we use this model, what we found was that UCMS over a period of weeks is associated with a decrease in behavioral rhythm amplitude. So this is when we looked at activity, so this is in panel A. you can see there's a decrease with UCMS that actually persists through recovery. We also see a similar change in body temperature amplitude. And these correlate with behavior similar to what was seen in the human studies, where here we're using a test, a forced swim test, which is a measure of behavioral despair, or the open field, which is a measure of anxiety-like behavior. And the take-home message is that the lower the amplitude, sort of the more anxious and depressed-like these mice become, whereas greater amplitude is somewhat protective. We saw the same thing when we looked at rhythms, molecular rhythms in the SCN. So we used mice that had a recorder that's in the SCN that allows us to look at rhythms in places over time. And again, using these same types of behavioral tests to look at depression-like behavior and anxiety-like behavior, we found that rhythm amplitude in the SCN is directly correlated with this behavior, in that the animals that had lower amplitude SCN rhythms had more depressive-like behavior, more anxiety-like behavior than the animals that had higher amplitude. So in conclusion, I've showed you that clinical studies show that stabilization of circadian rhythms via behavioral therapy or light therapy can be therapeutic in the treatment of mood disorders. Depression and anxiety-like behavior in mice are directly correlated with amplitude of SCN rhythms. Mice with a genetic mutation in the core clock have a manic-like phenotype, which is normalized by both lithium and valproic acid. And valproic acid also seems to rescue their circadian rhythms. And now we haven't tested whether one is dependent on the other, but it's very interesting that this drug is able to do both. And again, potentially suggests that this mechanism is important. And I've also showed you that there are novel compounds that target the clock that can specifically amplify rhythms, maybe be a little more targeted than something like valproic acid, and maybe not have as many side effects and be more therapeutic. And these are potentially really exciting new candidates in the future for the treatment of mood disorders.